one more, drilling it into our heads about the difference between a genotype and a phenotype, because you will definitely have to know the difference. So a genotype is a person's specific genetic makeup. This is the type of genes that you have. Whereas our phenotype is going to be an observable characteristic that's coming from our genetic makeup. So our phenotype is actually going to be a little bit of our genes, like we saw before, having a blue eye gene or allele of a gene and the brown eye allele. That is what tells us what we're going to see in the phenotype. But the genes aren't the whole picture. So our phenotype is going to include our interpretation of the genes, of that genotype. But our phenotype also includes things like our environment. Our environment can also influence what we observe coming from our genes as well. So genotype is just what version of a gene we have, and the phenotype is going to be what we observe in the end, and that's going to be made up of our genes and our environmental factors. And if we want to look at some examples, genotype is our specific genetic makeup, yet again. It's present from birth, so our genotype isn't going to change as we get older. It's pretty much set in stone. So it's unchanging. So if we talk about you being wild type at gene X, or brown eyes allele at gene B, which was our example, that's our genotype. If we're talking about a specific version of a gene, then that's our genotype. Our phenotype are those observable characteristics, and phenotype can be altered. It can be altered by other genes. So sometimes we get some really cool things like multiple genes interacting, and we can have the environmental effects taking effect here. So a phenotype would be height. So if we wanted to look at an example for height as the phenotype, our genotype might be a gene that produces growth hormones. And so if you have a certain version of that gene, if you have an allele that lets you produce lots of growth hormone, then we're probably going to see the phenotype of you being very tall. But the phenotype isn't just all about the genotype. So even if you have a gene that codes for you to produce lots of growth hormones, if you're in an environment that doesn't actually give you the resources to make those growth hormones, maybe you don't have enough food in your diet, maybe you're missing a particular important chemical, then even if your genes say you should be tall, the phenotype won't match because of the environment. So phenotype's just a little bit more complicated, and we have to take some more things into consideration. And so if we're talking about how do genes work, this is going to be a little bit of what we've already said. So genes are coding for the production of proteins. Our genes are providing the instructions that let us turn that information into the proteins that we need. And proteins are complex functional molecules. So they're complex, there's lots of pieces to them. They are functional molecules because they serve a purpose, they have a function. They're going to be hormones that help you grow, or pigments that change colors of things. They're doing something. And these molecules are made up of amino acids. So a particular type of chemical that gets put together to be the proteins that we need throughout our body. So if you have taken biology courses, if you're familiar with how DNA gets turned into proteins, you know about the idea of transcription and translation and turning things from DNA into proteins through all those steps. But that is not the focus for this course. We'll leave that to a cell bio course because we can get so much deeper into how genetics work, and it's a very cool topic. I highly recommend it, but we'll keep it pretty low level for here. So when we're talking about these genes giving us the information for producing these proteins, it can actually work in a couple of different ways. So you can have a gene be activated or deactivated. So within a cell, you can turn on or turn off a gene. And that turning on and turning off a gene can increase or decrease the production of proteins. So you could activate a gene that causes you to make lots and lots of the proteins that that gene codes for. Or you could deactivate a gene so that you stop producing that protein, and so the levels drop. So based on other processes going on within the cell, you can block using a gene to make proteins, and that changes the level of the protein within that cell. And so changing those protein levels can change the functioning within and around those cells. So if you deactivate a gene that codes for a growth hormone, if you stop producing that growth hormone, then that cell stops growing. And anywhere that that cell might have been sending that growth hormone, that will stop growing too. So we can have this kind of cascade effect where you can cause a change here that causes a change in the proteins, that causes a change in the function, and that can spread pretty far, which is so very neat. And so this is what we've already talked about. So a new term here in that we're talking about somatic cells. Somatic cells are basically all of our cells that are not related to sex. So everything that isn't a sperm or an egg cell falls under the category of somatic cells. And we make that distinction because our cells will have 46 chromosomes in those 23 pairs, except for our sex cells. Because if we think about if you got one copy of your chromosome from mom and one copy from dad, but if you as the offspring have two copies, so we can look at it kind of like this, thinking back to the diagrams that we've already seen, in order to pass along only one copy of a chromosome, you have to then take your two copies of a chromosome and turn it into only. And that's what our sex cells do, is they half the number of chromosomes that we have. So in a sex cell, we're going to have 23 chromosomes instead of the 46. And that's, again, probably a little bit more information than you need, but just in case you're wondering why we specify somatic cells, that's why. And so we've already talked about the fact that with two different copies, which one do you see? And that brings us to that idea of having a dominant gene or a dominant allele, like our brown eyes allele. The characteristic that this dominant allele has, so blue pigment or brown pigment, will be displayed. So if you have a dominant allele, even if you only have one copy, it's always going to be seen. If you have a version that is recessive, one of our alleles is recessive, that characteristic doesn't show up unless the partner gene, the other allele, is also recessive. So that's like we said with the blue eyes. You only see a child with blue eyes if both parents gave a blue eye allele. So dominant is going to be seen all the time, and recessive is only seen if you don't have a dominant allele nearby. So that's the distinction here. But that's not always the case. Again, because we're keeping it simple, we're only talking about a clean cut. It's either dominant or recessive, and that only one gene is involved. But I can tell you that it gets way more complicated than that. You can have partial dominance, And there's lots of fancy words for that if you go into genetics and different ways of inheritance. And those are really, really neat to look at, especially if you start looking at the genetics of, say, plants. But again, I can't really go into that detail as much as I would love to. But what we can talk about is another special case that does seem to apply especially strongly when we're looking at behaviors. And this is what happens when we see polygenic transmission. And if we do our breakdown of this word, so poly is a root that means many, and then genic meaning genes. So polygenic transmission means that the transmission of a trait of what you're seeing as a phenotype is driven by many genes. And I like to break things down into root words because it tends to stick in your mind just a little bit better. But when we're talking about polygenic transmission, we're saying that a number of gene pairs contribute to or combine to create a single phenotypic trait. So our phenotypic trait is going to be that observable quality. And this diagram here shows one of the easiest to grasp concepts of polygenic transmission. And this is the idea of skin pigmentation. So if you have eyes and have looked around, you'll notice that people exist in a variety of different skin pigments. People can be very, very pale, or they can be very, very dark, or they can be somewhere in between. And this quality is actually genetically driven. So the phenotype is influenced by the genetics that underlie it. And so if we look at the bottom here, we're looking at the number of darker alleles. So this would be the number of alleles that help us produce a pigment that is darker, that would help darken our skin. So if we have 0 dark alleles, then we're saying that you have no versions of any of the genes that allow you to produce a pigment. Or you can look at the other side of it, where we have 6 darker alleles. So you have 6 versions of those genes that allow you to produce darker pigments. And so if we're having a range between 0 and 6 possible alleles, we can think about that as, actually, let me draw it out over here. So if we think of it as gene 1, gene 2, and gene 3. So if gene 1, you get 1 copy from your mom and 1 copy from your dad, gene 2 is 1 from your mom and 1 from your dad, and gene 3 is 1 from your mom, 1 from your dad, then that gives us the 6 possible places that you could get an allele for dark skin. So darker pigments would be, say, let's go with capital A, means that you have a pigment producing version of the gene. And my A's all look very, very differently, and I'm sorry about that. But this would be a case where we have 6 different alleles that are all coding for dark pigment. If, say, in this case, you didn't get a capital A to get a pigment, maybe you have a less functional version. Maybe you have a little a. So this one would be like the blue eyes. It's a different kind of pigment. Or in this case, a non-functional pigment. So something went wrong in the gene and it doesn't produce any pigment at all. So in this case, you have one non-functional allele that's not producing a pigment, so you only have 5 times the pigment, which would be like this bar here. So your skin tone is darker, but not as dark as it could have been. And so as time goes on, or looking at different people, maybe they had a couple of times where they have these non-functional alleles, and we start seeing sort of closer to a medium skin tone. So a couple of darker pigments and a couple of non-functional pigments mean that you only produce half as much pigment as you could have if you had all six, and so on and so forth. So you could eventually get to the point where all six of them are not functional and your body doesn't produce any pigments at all. And then you have the palest kind of skin. You have the least pigment because you don't have any versions, any alleles at these locations that produce pigment. And so that's how we can look at multiple genes, it being polygenic, to transmit this phenotype. So these interact all together to give us our overall phenotype. And on that idea of sort of larger scale concepts, we can also look at epigenetics. And epigenetics are a lasting change in gene expression that are occurring through development. So they're not specifically caused by the genes themselves, they're caused by other factors, things that are external or environmental. So if things are outside of your body, they're part of your environment. So if we talk about external factors, we're talking about environmental factors. And so if you start looking at the epigenetics of a particular trait, you're looking at what else other than your genes is driving that particular trait. So if we're looking at height, you might have that gene that says you should produce lots of growth hormone and be really tall, but if your environment doesn't provide you with the stuff that you need to grow tall, then that epigenetic factor, that environmental factor, is changing your phenotype from what we'd expect. So just differing expressions here.